All right, welcome to The Explainer. Today we're unpacking a monumental, totally hidden process. We're talking about the epic journey every single medicine takes from being just a concept in a lab all the way to sitting on your local pharmacy shelf. It's this grueling marathon of brilliant science and immense resources. Literally, it's the hero's journey of a pill. So how exactly does a raw chemical actually become a cure? Just think for a second about the intense obstacles involved in creating those little pills sitting in your medicine cabinet right now. It is definitely not just, mixing some liquids together in a beaker. It's an incredibly highly calculated and honestly often really frustrating process. 10 to 15 years. Yeah, you heard that right. That is the shocking timeline required for just one single new drug to be discovered, tested, and ultimately approved. It's a massive undertaking, and it carries enormous commercial risk while demanding thousands upon thousands of hours from dedicated researchers. To really understand this epic obstacle course, we're going to trace the path right from nature to the lab, then move through drug discovery, preclinical testing, clinical trials, and finally, the regulatory verdict. Okay, part 1, from nature to the lab. To appreciate how complex today's medical pipeline actually is, we really need to start by looking at the origins of medicine. Historically, treatments were derived directly from nature. We're talking opium from the poppy plant, quinine from cinchona, or digitalis from foxglove. Today, however, the vast majority of new therapeutics are highly complex synthetic agents designed using some wildly advanced 3D molecular computer modeling. Which brings us to stage 1, drug discovery. This right here is the first major hurdle. It's an incredibly costly starting point where candidates are selected purely based on their pharmacological properties. When you map out the entire drug discovery and development pipeline, it is honestly just massive. Starting right at the beginning, you have these distinct phases, incredibly rigorous checkpoints, and a severe drop-off rate as the journey continues. Breaking down that timeline chronologically really reveals the scale we're dealing with. Discovery alone eats up between 2 and 5 years right out of the gate. Preclinical testing takes about a year and a half, and that is followed by a massive 5 to 7 years for clinical development. And even after all of that, regulatory approval still takes another 1 to 2 years. So what is actually happening in those first 2 to 5 years? Well, there are 3 crucial steps. First, there's target selection. This is where scientists identify a specific target inside the body and validate its connection to a disease. Second is lead finding, which is basically generating the potential new drug molecule itself. And third is lead optimization, where chemists meticulously tweak that molecule to increase its potency, selectivity, and stability. Now, you might be wondering, where do these leads actually come from? The sources are surprisingly wild. Beyond computer modeling, scientists still isolate leads from things like the venom of deadly krites and cone snails, various marine invertebrates, and even microorganisms like streptomyces, which, by the way, is exactly what gave us penicillin. Once a lead is finally found, scientists have to isolate its pharmacophore. That's just a term for the specific molecular groups that interact with a receptor to trigger biological activity. Think of it kind of like a structural key that scientists are trying to lock perfectly into place and then enhance during the optimization phase. But as you can imagine, this optimization process is notoriously difficult. There's this brilliant analogy from the source material that says lead compounds, like antisocial teenagers, refuse to give up their bad habits. I absolutely love that quote because it totally nails why lead optimization is often a painfully frustrating process for chemists. Despite all this ingenious and backbreaking chemistry, sometimes the molecules just absolutely refuse to behave safely or effectively. And because optimization is so incredibly difficult, pharmaceutical companies have to cast a really wide net. A typical pipeline begins with around 100 different projects. That's 100 potential paths explored simultaneously, just hoping to find a reliable starting point. Next up, stage 2, preclinical testing. Here, things shift gears from chemical tweaks in a lab to rigorous non-human safety checks. Remember those 100 initial projects we just talked about? Well, out of all of those, only about 20 compounds will actually make it to preclinical development. That means a staggering 80% of ideas fail before ever leaving the purely chemical discovery phase. So, the surviving 20 compounds now enter a rigorous year and a half of non-human testing.
This involves screening for acute and chronic toxicity, running mutagenicity tests, like the Ames test, on tissue cultures to make sure the cells don't mutate, and conducting extensive reproductive safety studies. Let's move to stage 3, clinical trials. This is the longest, easily the most expensive, and definitely the most critical stage. Actual testing in human beings. The funnel gets even tighter here. Our 20 preclinical compounds drop down to just 10 candidates entering phase 1 of clinical development. By phase 2, we're down to 5. And by phase 3, only 2 candidates remain. The attrition rate is just staggering as the scrutiny increases. So these clinical trials are systematic, experimental, biomedical studies in humans. Over 5 to 7 years, phase 1 starts by evaluating safety in healthy volunteers. Phase 2 assesses efficacy and dosage in small patient groups. And finally, phase 3 involves massive controlled trials to evaluate overall effectiveness and any side effects. Actually, let's pause for a second, because modern trials are seeing a massive shift toward biopharmaceuticals. These are therapeutic agents produced by biotechnology, like recombinant DNA, rather than conventional synthetic chemistry. They use things like vectors, such as viruses, to clone and express genes, which allows us to address complex hereditary diseases, but requires entirely new trial methodologies. And that brings us to stage 5, the final verdict. This is the finish line of our journey, where a literal mountain of gathered data is finally submitted for regulatory approval in hopes of actually reaching the market. 1. After 10 to 15 years of work, starting with 100 projects, dropping to 20, then 10, 5, and 2, we end up with one single approved drug. A full decade of financial investment and backbreaking science basically comes down to a 1% success rate. But you know what? The journey doesn't even really end with a regulatory yes. There's phase 4, post-marketing surveillance. Once a drug is marketed, its safety, long-term toxicology, and effectiveness still have to be monitored out in the real world, across the general population, indefinitely. Which brings us to our final thought for today. If it takes 15 years and literally hundreds of failures just to cure one single disease using these traditional pipelines, what revolutionary new methods are we going to need to conquer the rest? Whether it's through advancing molecular modeling or pioneering new genetic medicine, the entire future of the pharmacy shelf really depends on it. Thanks so much for joining me on this explainer, and keep learning.